Now, once the cortex and or the amygdala perceive danger in the environment, then the next step is to activate the locus coeruleus. And this is in the brain stem. And this has sometimes been referred to as the adrenal gland of the brain. It's sometimes been referred to as the adrenal gland of the brain. Okay? And in a sense, this is kind of the launching pad for normal fight or flight responses, but it's also the launching pad for anxiety disorders. Okay? Here's where you have about 40,000 norepinephrine cell bodies. And in terms of brain neurons, that is a tiny, tiny, tiny percentage. It's not necessary to know this, but it represents 1/10,000th of 1% of brain cells are these 40,000 norepinephrine cell bodies, okay? And they're right in this little tiny area in the brainstem. But they project widely up into other parts of the brain, okay? And so when there's danger in the environment, it activates locus coeruleus and it immediately spritzes the brain with norepinephrine, and which basically does two things, okay? One is it brings the person, or it could be any mammal, into a state of high arousal and vigilance, okay? Which is obviously very important if you're in potential danger. It's not a time to take a nap or be drowsy. Now, I've told this story before in the biology class, but I got to tell it again because I like this. It's a metaphor, but it's true. And they've done a study with animals where they put an electrode in locus coeruleus, and then when there's something in the environment, that's potentially dangerous, it starts firing like crazy, and it can register on the electrode. So for most people in a class like this, a day-long class, which happens to be long and exhausting, right? Okay. In the morning, if we inserted electrodes into your locus coeruleus, probably what you would hear is something like this. And what the locus coeruleus does when people are awake, and at least trying to be awake, it like a pacemaker kind of thing. It is periodically spritzing the brain with norepinephrine and keeping you awake. Actually, keeping you awake is an active process. Okay. If you want to increase locus coeruleus firing, one thing you can do is you can drink caffeine, and it'll cause locus coeruleus to fire more. Okay. Now, by afternoon, about this time in the afternoon, this is what you'd hear. Okay. The more the professor is boring, the slower the rate of locus coeruleus firing. Now, this is not due to having had a big lunch. Okay, it's partially due to getting worn out because you're tired, but there's a change in the circadian rhythm. And in primates, there's this dip in the afternoon, and there's decreased arousal. In monkeys, apes, and normal humans will not work. They'll take a rest, take a little nap, that sort of thing. But what if I say, it's like you're in your Psych 101 class, and you're about ready to slip into an irreversible coma. 2 o'clock in the afternoon, boring professor. And then you're sitting in the back, kind of scrunched down so he doesn't see you. And then he goes, "What do you think about this?" And then, okay, and that's exactly what will happen. Is you'll go from very low activity to a massive amount of norepinephrine spritzing the brain. Oh, what? Okay, that sort of thing. The other thing is it plays a role in activating, in a sense, kind of igniting the next level of stress response. Okay, now. Again, looking at this just from a pharmacological perspective, we can intervene at the level of threat appraisal. The other level of intervention is to go down into the brain stem and try to reduce excitability in the locus coeruleus. Okay? There are 4 major types of inhibitory receptors on the surface of these norepinephrine nerve cells. Okay, let's start down here. Serotonin receptors. Now, you may remember from the first part of this class that there are 15 different serotonin receptors, and it's believed that 14 of them are inhibitory and 1 is excitatory. So these are inhibitory serotonin receptors, and when they get turned on, okay, if there's increased serotonergic activity, then it will reduce the likelihood of the cell getting turned on. Okay, it'll help shut down the reactivity of this. And the quickest way to do that, to jack up serotonin, is to exercise. When people are very, very anxious, one of the best things you can do is for 10 or maybe 15 minutes is jog, run, or take a brisk walk. Almost without exception, there is a noticeable decrease in anxiety. Now, people still may feel quite anxious, but they actually feel less anxious, okay? So this is a very rapid way to do this. Ultimately, if you use drugs like SSRIs and other antidepressants that increase serotonin, it'll have an effect, but that takes longer to kick in than exercise. Okay. Opiate receptors, found in many parts of the brain. They are designed by nature to respond to endorphins and enkephalins. And one thing that's interesting is that when you rock mammals, it stimulates nerve cells in the cerebellum that cause the release of beta endorphin into the brainstem, into the locus coeruleus. That's why you can take a kid who's crying and they're out of control crying, and you rock them, and within a minute they start calming down.
What did you say it releases?
I beg your pardon?
What did you say it releases?
It releases endorphins, okay? Naturally occurring morphine-like substances. And it's always been just amazing to me how you can get a wildly upset, crying child, and in a minute or two they calm down. Well, they're getting like a little hit of heroin, basically. I mean, why do people like rocking chairs? Okay, if you look at people in the immediate aftermath of some horrible trauma, like a natural disaster, a lot of times you'll see them rocking because they're just doing it instinctively, because it dampens a little bit, anxiety. Okay, or a kid, just get real upset. Here, come here. It'll be okay, and you'll be okay. Why do people do that? Because it works, okay? Now, a very powerful way to reduce excitability in locus coeruleus is take heroin. Now, I don't recommend that you suggest this to your patients, but some of them will do it anyway, or prescription pain medicine abuse, like OxyContin and Vicodin and codeine and so on and so forth, all activate opiate receptors big time, okay? Hang on just a minute, I want to see which slide I have. Okay. Now, this is next is GABA benzodiazepine receptors. So let me show you how this works. On the surface of the majority of nerve cells in the brain, not all, but the majority of nerve cells, not just in the brainstem, although we're talking here about just in the brainstem locus coeruleus, okay? You have GABA benzodiazepine receptors. All right, now here's what happens. These are ion channels. They're receptors on ion channels. So this is the inside of the cell. This is the outside, okay? And what there is a bunch of negative ions. This is work. Okay, a bunch of negative ions floating around in here. And they are attracted to the interior part of the nerve cell, but they can't get through the cell membrane except through an ion channel, okay? And these particular negative ions are chloride ions, okay? And they have a slight electrical charge to them, okay? Negatively charged. They can't penetrate into here, so back. Oh, come on. Because the channel is closed. Now, from our first class, I believe I've talked about these ionic receptors on ion channels. The ion channels are these proteins that if you tweak them the right way, they'll move, and they transiently open, and then ions can come in or out, depending on what kind of channel it is. And so what happens, now wait just a minute, this is out of control. What did I do? It's intentionally trying to review the material. Okay. Right. Anyway, so what happens naturally then is this is an amino acid that is made in the brain, GABA, and it's gamma-aminobutyric acid, but nobody ever says that because it's a mouthful, so everybody calls it GABA. You might remember it's the most common inhibitory neurotransmitter in the human brain from one of our early classes. So what happens then is when GABA is released, here we have a GABA molecule and it's moving towards the receptor and it binds and it opens up the ion channel. And then you have an influx of chloride ions and the cell goes, it doesn't actually do that, but it actually, it hyperpolarizes it, which means it makes it less likely to get turned on. Okay, it's like shutting it down. Okay, now this lasts for a microsecond, and then it'll knock off the molecule, and the channel will close again. But then along comes another one and opens it up, and so this is continuously happening in hundreds of trillions of these kind of ion channels every moment in the brain. Now, there are also binding sites on these ion channels for benzodiazepines, which are tranquilizers, but also alcohol binds to the benzodiazepine receptor. Okay, so there's a lot of similarities between alcohol and tranquilizers. This is one of them. There are other binding sites where barbiturates can bind to this. So there are several different kinds of receptors on these ion channels, and they can work together. Okay, so GABA by itself can cause the opening of an ion channel. Okay, at least to some degree. But if you take a tranquilizer, okay, now watch this. Here, somebody took, a Valium or a Xanax or something 45 minutes ago, and now we got it's up in the brain. And watch, this is like a Xanax moment. Watch this. See that again? Okay. Oh, yeah. How do you spell relief? Okay, so it actually helps to facilitate the opening of this channel, and you get more negative ions in, and you calm down. Okay, and so that's our GABA benzodiazepine receptor. Now the problem, once again, is that since this is seen on the majority of nerve cells in the central nervous system, if you take a bunch of Xanax, yeah, you'll calm down, but it'll put your brain to sleep, okay, your cortex. So it can interfere with alertness and cognitive processing and that kind of stuff. So that's one of the downsides of this. Now, if you want to get really, really relaxed, okay, you don't write this down. Okay. You don't even record this. No, I'm just teasing. Get a whole bunch of Xanax or something like that, Valium or Ativan. Put it in a blender, and then put in some vodka. Blend it up, a little twist of lime, and just have a good time. And what will happen is when you combine alcohol and a significant amount of alcohol and tranquilizers together, it really gets you relaxed, really relaxed. In fact, what it does is the benzodiazepine GABA receptors down in the brainstem area that controls respiration relax so much you stop breathing and you're dead. Okay? So that's the ultimate relaxation, right? Now, you can take a bunch of benzos and it will not kill you. You'll be asleep for 3 days maybe, but it won't kill you. But if it's mixed with alcohol, then the combination is very deadly, as well as mixing barbiturates with alcohol. And there have been many reported cases of people accidentally or intentionally using that combination, and they results in death. Now, the final receptor here, let's go back for just a second, is called the alpha-2 adrenergic receptor. Okay, and we have 4 classes. There's probably several hundred of each one of these on these norepinephrine nerve cells. And so here, come on, John. What's the deal here? Okay, now what's cool about this is here we have this norepinephrine cell, which means it produces and uses norepinephrine as its neurotransmitter. So you get a message from the cortex saying, oh my God, here comes trouble. It activates this and it fires off the nerve impulse, and this may go up to the hypothalamus or up to the cortex, or it goes somewhere. It goes widely distributed throughout the brain. But simultaneously, there are they're called collateral axons, and they're axons that come off of here, just like a normal axon, except that they bend back on the nerve itself. Come on. And so what happens then is at the same moment that it is activating other parts of the brain, it's squirting itself with norepinephrine. But instead of norepinephrine now being excitatory, it's activating the alpha-2 adrenergic receptor, which is an inhibitory receptor. In other words, throughout most of the brain, it's turning things on, but down here, it's activating these receptors that are inhibitory. It's putting the brake on. And this is cool, because that way you have immediate reaction, but if there's no danger, it'll shut itself down real quickly. So you almost get in a car accident and you have that kind of thing like that. That's the sympathetic nervous system, okay? But if you determine a second later, oh, thank God, I didn't have a wreck, I'm not in any danger, then you start to calm down. Now, you'll be shaky for a while because you've got to metabolize and excrete out some stress hormones that got dumped into your system in that half a second, but you'll calm down fairly quickly. So this is a self-regulating mechanism, okay? Now, let's go back. If I can go backwards here. Go back here. So alpha-2 adrenergic receptors. Now, norepinephrine turns that on, okay? If you take something that increases norepinephrine, it's going to increase it everywhere in your body, not just here. The entire sympathetic nervous system uses norepinephrine. So trying to take norepinephrine or amino acids precursors to norepinephrine don't work. Okay, but one way that you can selectively target that receptor and turn on the brake is by taking an alpha-2 agonist. Where an agonist turns something on, here it's turning on the brake. And that's a class of medications that was originally developed to treat high blood pressure. It's estimated that about 90% of cases of hypertension are due to anxiety or stress. And so one way of dealing with that then is you go right down to like the launching pad and you just turn it down, shut it down. And the most common drug that's used that operates on the alpha-2 adrenergic receptor is clonidine, also known by the brand name Catapress. Lowers blood pressure. And we'll talk, we're going to, next time we get together, we'll talk about the treatment of ADHD. And we're going to see that the clonidine is not infrequently used in the treatment of ADHD. Now, the good thing about clonidine is that, and it's like serotonin and also clonidine, is it quiets the activity at the locus coeruleus, but it leaves higher brain regions awake and alert. It's kind of the ideal, in a sense, anti-anxiety drug because you don't want to put people to sleep or impair their ability for normal reaction time and so on and so forth, but on a biological level to reduce the intensity of the anxiety response.